Today we're going to talk about the urinary system. We'll start with the function of the urinary system, to get rid of waste and to maintain extracellular fluid. Also, we will talk about evolution, how the kidney developed, different parts of the nephron, the functional unit of the kidney, and how it interacts with the vasculature to filter the blood and to return important components back to the blood, as well as excreting the waste products of the body. Also, we'll talk about the excretory organs, go through the ureter, the bladder, and the penis for the outlet through the urethra. We'll talk about the transitional epithelium that's found there only in the urinary system. If this is useful in your learning of medical histology, please share with a friend. Thank you. Medical School Histology Basics. Urinary system. Hi, I'm Larry Johnson. I'm a professor at Texas A&M University, and today we're going to talk about the urinary system. We'll start with evolution, function, anatomy, the components of a nephron, how to identify those components, and excretory components of the urinary system. Now, the function of our urinary system is one of maintain homeostasis. It wants to rid the body of waste, urea, uric acid, creatinine, and salts, and to preserve the constancy of the extracellular fluid and composition, pH, and volume. It also has an endocrine function. It secretes erythropoietin for production of red blood cells. It also secretes renin for aldosterone release. Now, those functions are brought about by four basic mechanisms. One is filtration, and the filtration occurs in the renal corpuscle. Here we see the glomerulus and the renal capsule, and you see many of them all around. Active secretion, where they're secreting things into the tubule, or passive diffusion, where some things just go in and out based on the concentration gradient, and selective absorption, like proteins in the proximal tubule. And here you can see the proximal tubule, the loop of Henle distal tubule, and the first part, the proximal tubule, is where good stuff has to be returned. Sugars, amino acids, proteins, vitamins, and then the other portions are related to concentration of the urine with aldosterone influence and ADH, and finally in the collecting tubules. And paralleling the nephron is the Peritubular Capillaries that follow along even down into the medulla. So in terms of evolutionary, whenever animals wanted to escape predators, they first of all were living in the sea, and that's what we see here in the yellow. And then they moved into brackish water. When they moved into brackish water, they had plenty of water, but they had to preserve salt. And that's where we have the development of the glomerulus. So the glomerulus was a capillary tuft that was embedded into the wall. And then when the animals proceeded to be amphibians and walk upon land, not only did they have to preserve salt, they also had to preserve water as well, and that's where we developed a long loop of Henle. Some of the animals have returned to the sea, some have lost their glomerulus, some still have them, but land animals have this typical makeup where you have glomerulus here and then a long loop of Henle. And so you can see the early nephron, which was just an opening, and the glomerulus was associated with that opening, and then finally the glomerulus was embedded into the wall, as you see there, and also the blood vessels flow. So here we can see where the glomerulus is embedded into the wall. So this is the wall of the nephron that goes around here, and we learn that these are podocytes, and the podocytes and the parietal epithelium, both of those are actually part of the original wall. And so we can see that this is the parietal epithelium here, the podocytes in through there, and the blood capillaries in through here. as we can see. Now another thing happened is the blood flow changed. See, the blood flow's coming in up here, and then it came in from different directions. Then the blood flow changed such that it only come from in the glomerulus layer and through there. So it goes, and this made a portal system. So we have portal arteriole, the afferent and efferent arterioles. And if you remember when we talked about portal systems, you had to have two capillary network and a portal vessel in between. And that's what we have here in the kidney. The first capillary network is the glomerulus, and the second capillary network is the peritubular capillaries. So first capillary network, second capillary network. So you see the afferent arteriole, the capillary network, first capillary network, and then the efferent arteriole, and then the peritubular capillaries. And remember, the portal system causes a local change in blood composition whereby the first capillary network modifies the blood and the second capillary network benefits from the changes by interacting with cells nearby. So the first capillary network is the glomerulus, and the second capillary network is the peritubular capillaries. And we'll revisit the peritubular capillaries. Here you can see the peritubular capillaries go around the nephron as well as the loop of Henle. So in the kidney, we have the cortex and the medulla, and you have the pelvis that is continuous with the ureters to the urinary bladder. So the main part of the urinary system is the kidney, ureter, urinary bladder. We also have urethra going out. And so if we look at the kidney, we see a cortex and a medulla. So this is a cortex and a medulla in through here. And the way the blood vessels go is that blood vessels create these little arcuate arteries. This is an arcuate artery that comes in through there. So here's an arcuate artery. It's a little arch because it goes over the pyramid. This is a renal pyramid, pyramid shape with a point over the top at this place where it empties into the renal pelvis. And then off from there you have little arteries that go up in here. We call those interlobar arteries, and that's what we see here with a little glomeruli going off of each of those. And those are off the arcuate artery, which comes in the interlobular artery. Interlobular, arcuate, and then the interlobar arteries is where you find the glomeruli in the cortex. And so we can see the cortex and the medulla, and here's a nephron sitting here with the glomerulus up in the cortex area, and then the fluid goes through minor calyx, major calyx, to the pelvis, and to the ureter. Now, where the ureter and the blood vessels go in and out, that is the hilus region of the kidney. And here we see the kidneys of various kidneys. We see the cortex and the medulla, and the cortex, medulla, and also we have a capsule that surrounds the kidney, dense irregular connective tissue with fibroblasts. We see the capsule located in different places. It's the renal capsule. You will see the capsule around this drawing, but with cortex and medulla with the capsule, and this is really a minor calyx that fluid is removed from this area. And we can see that on this Toulidine blue slide too. This is the cortex and the medulla. So cortex, medulla again, and what's located in these are nephrons, and nephrons are of different kinds. You have the juxtamedullary nephron, the one right next to the medulla, and those are the ones that have long loop of Henle, and those loop of Henles are located down deep into the medulla. And then there's also cortical nephrons. The cortical nephrons are located in the cortex, and they have a short loop of Henle, and the loop of Henle itself could be somewhat down in the medulla, or it could be in the cortex itself. Now, regardless of which type of nephron the juxtamedullary nephron or corticonephron, the collecting tubule in both cases goes down through the medulla and empties out here in the minor calyx through the papillary ducts that are there through there, papillary ducts. And so regardless of type, they still have to go through the medulla to concentrate the urine. But here you can see where the thin portion of the loop of Henle is located, the cortex for the corticonephrons. And here we can see in this one slide, here you can see the dark glomeruli, the glomerulus here. These are the corticonephrons and the juxtamedullary nephrons, of which we really can't tell any difference much. Later on, we will show a little difference because these are the ones that produce the renin that are next to the medulla with the long loop of Henle. But here we can see the thin portion of the loop of Henle in the cortex. See where we are up there, way up here in the cortex with a thin portion of the loop of Henle, as we said was on the corticonephrons. Now, we mentioned before about the afferent and efferent arteriole and the two capillary networks, but also these connect with the nephron. Nephron is a structural and functional unit of the kidney, and we can see it has the corpuscle, which is composed of Bowman's capsule, and then the tuft of capillaries is the glomerulus that's in there. That's the first capillary network in its portal system. And then from there you got the proximal convoluted tubule, and then you have a descending portion of the proximal tubule, a thin loop of Henle, ascending portion of the distal tubule, and then the distal convoluted tubule, and that empties into a collecting duct. And then the collecting duct goes out the pelvis through a papillary duct. So basically, the nephron consists of a glomerulus, which is how the blood interacts with the nephron, or how the filtrate is produced, the proximal convoluted tubule, loop of Henle, distal convoluted tubule, then the collecting tubule. Those are the components. And here we can see there's a difference in the afferent and efferent arteriole. The afferent arteriole is larger than the efferent arteriole, and that produces a pressure head that will help produce the filtrate. So remember here we had the afferent, efferent arteriole, and the capillaries of the glomerulus is in the first capillary loop of the portal system. And if we looked at these, we see the corpuscle. This is the corpuscle with the glomerulus inside. We see the proximal tubules that has a brush border that we can see here. The brush border that we see now, the brush border proximal tubule. And also we have a distal tubule. Distal tubule right here, we can see one. And then the loop of Henle is these portions here. We can see the loop of Henle. And then the collecting tubule is this here one with distinct boundary between adjacent cells. Similar type thing with H&E. We're in the cortex here. We can see the glomerulus. We can see the Bowman's capsule, the parietal cells that goes around here. Proximal tubules with mostly the lumen is partly closed because of the brush border that's in there. Then we have a distal tubule as opposed to a proximal tubule, another distal tubule. Collecting tubules with distinct boundary between adjacent cells, several of those. And then we have the thin portion of the loop of Henle. And we can see the glomerulus here is the tuftulary cuff of here, and this is the urinary pole where the blood vessels come in. The vascular pole where the blood vessels come in. Urinary pole is where Bowman's space can continue with the lumen of the proximal tubule. And then of the cells that we have the space here. We have the parietal epithelium, which makes the Bowman's capsule, so it's Bowman's space. And then we have visceral epithelium, which are really the podocytes that we talked about before, that also touches Bowman's space. Then we have a afferent arteriole, juxtamedullary cells in through here. Macula densa is part of a distal tubule, and this together is a renal corpuscle, and then Bowman's capsule is where we have the space there. if we want to see those again, we can see this is the glomerulus here and here, and we have the Bowman space, the space in between there, and then we have the parietal epithelium that surrounds this, in this case, and then we have the visceral epithelium, which are the podocytes. Remember, that was part of the original wall. Here we see the macula densa and the distal tubule, and this is our arterioles. So the arterioles produce a filtrate, glomerular filtrate, and it's brought about by the arterioles entering and leaving. You got the glomerular endothelial cells, so the blood has to go through the endothelial cells, through the basal lamina, through the visceral leaf to get into a Bowman space. So you go from the blood vessel through the endothelial cell, through the podocyte on the outside, to get in through there, and there is the basal lamina is in between there. So the barrier is between the blood endothelial cells, which are fenestrated, basal lamina, and then the podocytes on the outside to get into a Bowman space. So here we can see the different components of the filter. You got it fenestrated, and you can see the fenestra right here. This is the endothelial cells, fenestrated endothelium. You also have a basal lamina. It's a combination of a basal lamina between the endothelial cells here and the podocytes there. And then you have the filtration slits of the podocytes. The podocytes has major processes and secondary processes, and then it has little finger-like coverings that covers over the glomerulus. So you really can't see the capillary tuft with a scanning electron microscopic view. If you're looking in a Bowman space, all you see is the podocytes wrapped around each of these, and they have these little slits with a diaphragm for slit membrane as part of the barrier. So the glomerulus basal lamina is thick, ionic. It filters micromolecules according to size and charge. Now the glomerulus has extreme filtration, much more than others because it's large in size. And here you can see the glomerulus with the capillary tufts, and you can see here many glomeruli. It also large resistance afforded by the reduced diameter of the efferent arteriole. So the afferent and efferent is smaller. That produces a pressure head in the glomerulus for the fluid to go through to produce a filtrate. Thin filter, very thin. Endothelial cells, podocytes, and the basal lamina. And then also, here we see the fenestrated endothelial cells of the capillaries, so that it's 25 times more permeable than regular capillaries for the filtrate to be produced. So the hydrostatic pressure brought about by the difference in the size of these vessels produces 180 liters of filtrate a day. The filtrate resembles blood without large molecular weight, that is, those that are larger than 40,000 molecular weight. So if we look at the renal corpuscle, we see the urinary pole is over here, the vascular pole is over here, where you see the site of the afferent and efferent arteriole coming in there. The urinary pole is a site of union of the podocytes of the capsule with the proximal tubules cells. And here we can see that this is the visceral, this is the vascular pole. You can see the vascular pole here. This is the distal tubule, and then these are the artery coming in through there. There's a little arteriole coming in through there. Then we have a urinary pole. You can see where this has been removed, and this is going to be the proximal tubule down there. So this is these are parietal epithelium in through there, parietal epithelium right in through there and here, and this is where you have the urinary pole, where parietal epithelium gives rise to the cells of the proximal tubule that you can see there. And so here we can see again the macula densa of the distal tubule, juxtamedullary apparatus in through here, juxtamedullary cells in through here, distal tubule, proximal tubule, and you can see the microvilla of the brush border of the proximal tubule. And so on the proximal tubule, we can see that it has microvilli projecting. It actually has a uniform microvilli, which makes a brush border. And this is involved in absorption of protein, which is what we see here. Protein gets caught in there, and then it's phagocytized and broken down to amino acids to be reused. So we can see the brush border here. So all these are cells of the proximal tubule. You can see the brush border with the PAS staining, as well as the basement membrane, but the brush border is also PAS positive. You can see the brush border here, and there. And so there could be a little bit of protein in the urine, but most of it should be out. And so how do you identify the proximal convoluted tubule? One is by the brush border. You can see the heavy purple here, which is the brush border. You can also see a brush border in this guy as well, and of course at the electron microscopic level, brush border we can see there, microvilli. Also the cytoplasm is more red, and you can see red here compared to this distal tubule, not quite so red. And also the nuclei are far apart, a long ways apart. Nuclei are in the proximal tubule, and the boundaries are indistinct. And here you can see why. Scanning electron microscopic view showing that one cell interdigitates in another cell, and as a consequence it doesn't make a distinct boundary between adjacent cells. That's how you identify the proximal tubules. We see lots of proximal tubules in through there. Most of the tubules in here are going to be proximal tubules. And you see the nephrons, juxtamedullary nephrons. These are the arcuate arteries that go in through there. Medullary rays run up where the cortical material runs into the where the medullary material runs into the cortex. These are medullary rays where the nephrons are going down to the medulla to come back up to the top. Now, where the proximal tubule descending portion ends and the loop of Henle begins, you can see the transition. And so you see the brush border of the proximal tubule, and all of a sudden it becomes very thin. And you can see those in these specimens. Over here you have descending portion of the proximal tubule. Over there you got the thin loop of Henle. So we have several of those transitions that can occur, and that's where we are, right up here between the loop of Henle and the descending portion of the proximal tubule. You can see it here, here, but also you can see it when it comes back up. You can see a transition between the two as well, where this is the distal tubule and this is the thin portion of the loop of Henle. So what it appears is that the descending portion of a thin portion loop of Henle looks like this, but the ascending portion is more bubbly, like that. So this is a thin, this is the ascending thin loop of Henle, and this is the ascending thick distal tubule that we see. So we can see that again. So we're talking about a transition of the distal tubule here now. This is definitely distal tubule here, and thin portion of the loop of Henle. Same type thing here. You can see where the mitochondria are lined up by the infolding of the plasma membrane in the distal tubule. Also, it has no brush border. This is plainly distal tubule, and that is continuous with this thin portion of loop of Henle, which is bubbly, as we mentioned before. And so the descending portion looks like this, but the ascending portion, the cytoplasm is kind of bubbly. So this is the distal tubule That's ascending. You see the mitochondria lined up in through there. It has microvilli, but no brush border. And then we have a distal convoluted tubule. How do you define that? One, no brush border. Also, less eosinophilic, more less red than what you see in a proximal tubule. Proximal tubule, and here's a distal tubule. Nuclei are close together, as you see there and there, and the lumen is fairly larger. And so those are some of the characteristics you can see with the distal tubule compared to the proximal tubule. Proximal tubule has brush border, no brush border. Nuclei far apart, nuclei close together. Less eosinophilic. That's what we can see. And then where the distal tubule comes back up with a vascular pole, this is afferent, efferent arteriole, you have a high density of cells there. We call that macula densa. And this is an osmotic sensor. This macula densa senses osmotic pressure. It should be hypotonic. If it's not hypotonic, it will cause these smooth muscle cells, modified smooth muscle cells in the afferent and efferent arteriole to release renin. And so here we can see the arteriole. There's a distal tubule right in through there. It's kind of ragged, but it is. And if you look again, this is the, here we can see the renin in itself. So this is the renin that we see in the vascular pole right in through there, which are in the smooth muscle cells, modified smooth muscle cells of the afferent arteriole, and next to, right next to the macula densa. And so as we come, we see there's a rich vascular supply, not only to the glomerulus, but also in the peritubular capillaries. And they're all around in the cortex, as you see there, but also they extend down into the medulla as a vasorectae. And so we can see little groups of tubules, and you can see these are descending portions of loop of Henle we talked about before, but the thinner one, see how thick that loop of Henle is, as opposed to this real thin one here is the capillaries. And so we can see the capillaries in through there, and these are the capillaries that wrap around the proximal tubule as well as the distal tubule. This is down in the distal, this is down in the medulla, down in through here, but also in the cortex and the medulla, the peritubular capillaries surround them. And so we can see the similar type thing. So we see the peritubular, we see the peritubular capillaries coming out from the efferent arteriole, going down various lengths down in through there, and you can see the renal corpuscle here, and you can see the nephrons going in through there with the peritubular capillaries wrapped around it. And here we can see the vasorectae. Blood enters at the top at 300 milliosmoles, which is normal osmotic pressure, and it also leaves with 300 milliosmoles. But meanwhile, as it was going down, it increased the concentration of salt and urea increased as it went down deep into the medulla, and this is known as a countercurrent exchanger. And so basically it goes down, and the osmotic pressure changes within there, but it's reduced again as the salt goes out of here back into there, so that it doesn't disturb the gradient that was set up by the thin loop of Henle of the nephron, and that is called the countercurrent multiplier, is the nephron itself. So here we see the peritubular capillaries. About 180 liters a day is filtered. That's 4 times reabsorption of all of the vessels in the body. The endothelial cells are really extremely porous. They're fenestrated. The osmotic pressure, because the proteins have been retained, that brings the fluid back in. Low capillary pressure and proximity to the urinary tubules. So these are the glomeruli, the urinary tubules, but then the nephrons, and then these are the peritubular capillaries. About 85% of the water sodium of the glomerular filtrate is reabsorbed by the proximal tubule. So the bulk of this 180 liters never gets to the medulla. It's already absorbed from the proximal tubule. And from the distal tubule, you have a collecting tubule. And the collecting tubule is distinct by distinct boundary between adjacent cells. And the cells are getting taller, maybe more columnar, as you can see. So this is a thick and thin portion of a loop of Finley, and this is a collecting tubule. And when you get down, the collecting tubule becomes even taller and very distinct, and it becomes a papillary duct that empties into the minor calyx. Here we can see the tall cells of the collecting tubule. And so we have the capsule, of course, around the outside, and then we have the minor calyx, where you have transitional epithelium located in through there. So the papillary ducts that we saw right here, they give rise to transitional epithelium. Again, we can see the minor calyx, so it's medulla coming down, be transitional epithelium right in through there. So you have these papillary ducts opening up, transitional epithelium. Transitional epithelium will be in here and here. This will be the minor calyx, and then it goes with the major calyx, then it goes through the pelvis, and then it's out through the ureter. And here we see a ureter. Ureter has transitional epithelium, it has a lamina propria, and it has a muscle coat. And it's got the intercircular outer longitudinal muscle, and it has an adventitious attachment as well. So this is the ureter. Then we have the urinary bladder, transitional epithelium with muscle again. We can see one here. Similar type thing, lamina propria, transitional epithelium on the lumen, muscle coat, smooth muscle of course, and adventitious. Now what happens is that the transitional epithelium that we see here has to transform itself whether the urinary bladder is full or empty. If the bladder is full, then they're stretched out. But if the bladder is empty, then they fold back on themselves. There's a rigid part. Proteins get together and they make the membrane rigid, like we see here, a rigid part. And that makes, when it folds up, it makes little flattened vesicles. You can see these flattened vesicles here. These flattened vesicles are the same thing. So this is continuous with the lumen. The cell itself can't stretch. What it does is the plasma membrane folds up and then it unfolds, and that's facilitated by these rigid portions of the plasma membrane that makes these little flattened vesicles. And then if we follow the urinary tract out, you have the urethra in through there. In the case of the penis, here we see the urethra could be transitional epithelium on there as well. So again, in the proximal tubule, you restore the salt and the water, 85% of it. Protein, that's in the proximal tubule. In the loop of Henle is where you set up a concentration gradient that you can then benefit with aldosterone and ADH to concentrate the urine to remove 99% of the 180 liters of fluid that was filtrated out of the glomerulus in the beginning. So in terms of function of the urinary system, it maintains homeostasis, gets rid of waste, but it also maintains the extracellular fluid balance and composition, pH and volume. It also produces erythropoietin. Erythropoietin is produced by the interstitial cells in the kidney, and then renin is produced by those modified smooth muscle cells in the afferent and efferent arterioles, and that's responsible for aldosterone release. So we have a couple questions. Which of the following process structures combination of the urinary tract match? Filtration, renal corpuscle? Yes. Reabsorption protein, proximal tubule? Yes. Reabsorption of sugar, distal tubule? No. It's already done in the proximal tubule. So the answer D.
Renal corpuscles facilitate interaction between blood and urinary tubules. Yes. Contain parietal cells that filter blood? No. The parietal cells contain the filtrate. It doesn't touch the blood. Contain mesenchymal cells that has renin granules? No. The renin granules are located in the smooth muscle cells of the afferent arteriole. A is the answer. Which characteristics facilitate maximum filtration of the glomerulus? Large filter? Yes. Thin filter? Yes. High blood pressure created by a difference in the caliber of the afferent? Yes. The answer is E. A, B, and C. We want to thank the various books from which we were able to get some pictures from, diagrams from. Thank them because they are the sources of the material other than the histologic specimens that I have. This is where one time I went to Japan, and this is Mount Fuji. You can see this is from my hotel view, and we went all the way up to the snow line. We were right up there at the lodge at the snow line on Mount Fuji. This is the end of Medical School Histology Basic Urinary System. Thank you. And if this information was useful, please share it with your friend and colleagues so that it can help them with this free information as well. Thank you.